think it's time, right? So, Dr. Greg Nacarelli, and all sorts of letters after your name: ND, PhD, MS, CNS, CNGS. I don't even know what those are. Is passionate about advancing the field of integrative medicine. He embraces cutting edge data analysis practices. He earned a doctor of naturopathic medicine from Bastyr University in California, master's degree in individual genomics and health from Johns Hopkins, a PhD in nutrigenomics, fatty acids, and cardiovascular disease and healthcare, genetics and genomics from Clemson University. He's the director of metagenomics for the American Board of Precision Medicine as a precision health consultant with Theriome and Self Code. Dr. Nacarelli is an expert in integrative precision health and lectures on integrating genomics, metabolomics, metrogenomics into integrative medicine practice. Did I get them all right? Did I get that right?
Yes.
Those words? Not too bad.
Close enough.
His goal, I never even heard of those words, 10 years ago.
Yeah.
His goal is to facilitate the adoption of omics technologies into healthcare for improving diagnosis and treatment. And, as we always say, we will be smarter by the end of this lecture than we are now. So, Dr. Nacarelli, thank you so much for being with us, and take it away. The floor is yours.
Absolutely. Thank you so much, Dr. Clearfield, for the intro. And yes, the CNS is there's an American Nutrition Association in the United States. It's one of the two big nutrition organizations. They have a board certification for nutrition, and that's what the CNS is. And then they also have a nutrigenomics certification, and that's what the CNGS, Certified Nutrigenomic Specialist, is, just to clarify. Okay, so I'm going to share my screen. All right, so I will be covering metabolomic pathways and metabolic syndrome. And so I've already had the introduction. I'm a naturopathic doctor and a PhD in genomics. I'm a precision health consultant at Theriome. My goal ultimately is to combine natural medicine with omics technologies, ultimately to improve precision and personalization of medicine. And so for today, I'm going to be covering two major topics. I'm going to be talking about metabolomics and then talking about metabolic syndrome. Within metabolomics, I'll be covering what it is, how it's associated with systems biology, some core metabolic pathways, looking at metabolic rate, some objective and subjective assessments that are relevant to metabolomics, some of the benefits and limitations versus with metabolomics, and then a metabolomics-centered patient visit. What does that look like? And then I'll look at metabolic syndrome, look at the pathophysiology, how mitochondrial dysfunction plays a role in metabolic syndrome, and we'll look at metabolic perturbations in metabolic syndrome and also what you can do with it in terms of interventions. So what is metabolomics? Metabolomics is the study of metabolites present in an organism. You can look at various different tissues to look at this in terms of blood, urine, stool, saliva. There's metabolites found in all of those. We can make health predictions by looking at these metabolites, and these predictions can possibly impact treatment recommendations or preventative strategies, and ultimately they're going to be impacting the phenotype. So metabolomics is systems biology. So traditional medicine, looking at things individually and not how they're associating with each other, is reductionistic in nature. And when you look at systems biology, which is essentially system science, it's looking at the relationships of things to each other, looking at relationships of organ systems to each other, looking at the relationships of molecules and their relationship with each other. And then there's multiomics, looking at the proteome, which is an entire collection of proteins, or the metabolome, looking at the entire collections of metabolites. How are they associating with each other? That's more so systems biology, and this is kind of where modern medicine is hopefully moving towards, because it's a more representative demonstration of what is actually occurring in our body. These systems are working with each other, not separate from each other. Some core metabolic pathways. Most of you probably have had biochemistry, so I'm sorry if I give you PTSD by showing these. I promise I won't go in depth with these pathways, but within these pathways are the metabolites that are being tested within metabolomics. And so core metabolic pathways: glycolysis, glyconeogenesis, the pentose phosphate pathway making NADPH, TCA cycle, that's a very big one. This is how our mitochondria is going to be generating energy. Subsequently, from that, oxidative phosphorylation. Fatty acid metabolism, synthesizing fatty acids, elongating them, oxidizing them, ketone body metabolism. One carbon metabolism, also famously known as the methylation cycle. Detoxification is a metabolic pathway. There's lots to look at there. I have an example on the right here of phase 1 versus phase 2 detoxification, and there's a bunch of metabolites that are associated with this. Neurotransmitter synthesis, urea cycle, nucleotide metabolism, heme metabolism, oxalate metabolism, hormone metabolism, cholesterol biosynthesis. These are all core metabolic pathways, and of course, there's others that are not mentioned here. We can look into two major categories with metabolism. Are we breaking down molecules or are we building them? And so some catabolic or pathways that break down things, glycolysis, pyruvate processing, TCA cycle, oxidative phosphorylation, beta oxidation. And then there's anabolic pathways. And so amino acid synthesis, the pentose phosphate pathway, fatty acid synthesis, we could put hormone synthesis in here. So there's a distinction there between the two. There's the rate of metabolism. We're familiar with some of these terminologies, but there's our basal metabolic rate. This is the minimum energy required to sustain life. This is at waking state, no food, no movement. Then there's our resting metabolic rate. So it's basically our basal metabolism plus sleeping metabolism plus arousal metabolism. And then our exercise metabolic rate. So this is metabolism during exercise plus our resting metabolic rate. You could look at different metabolite shifts at each one of these stages, and you'll see different metabolome signatures depending on which one is occurring. And I will talk about exercise metabolites a little bit later. We could look at the impacts of macronutrients on metabolic rate. So proteins are going to just have the highest contribution to metabolic rate, especially because they're going to be making enzymes and these other protein components that are going to be generating heat. And so they contribute a large portion of our metabolic rate. Carbs, maybe initially more of a contribution, but certainly not over the long term. And then lipids is a little bit more of a long view of contribution to metabolic rate. Everything is timed in our body. And so our organ systems are going to be metabolically active at different times. And so obviously we have our sleep-wake cycle. Different parts of our body are going to be active at different times. Various things can disrupt this, such as shift work, sleep deprivation, time zone travel. Aging may alter this. But we could look at different organ systems of when the stomach is going to be most metabolically active. When you look at the liver versus at midnight versus midday, you're going to see different enzyme pathways upregulated. Fatty acid tissue, you're going to see different, these genes here are coded for adiponectin, which is a compound secreted from our fat cells. We'll see different adiponectin genes upregulated or downregulated at different times. And of course, dysregulation of these pathways are associated with insulin resistance and metabolic syndrome. So this is where circadian biology is very important when it comes to metabolism and just metabolite testing in general. So general rule regarding metabolic pathways. So as I just mentioned, the time of day is going to affect these pathways. So make sure if you're doing a metabolite test that you're testing it at an accurate time. If you have all elevated metabolites or all decreased metabolites in a pathway, that's not good. If you have a high upstream metabolite and then a low downstream metabolite, that could indicate several things. It could indicate enzyme deficiency, such as genetic deficiency in nature, or it could be chemical inhibition that's leading to an enzyme deficiency, such as an environmental exposure, or there could be a low substrate or cofactor. For example, when we look at something like the MTHFR gene and lack of methylated folate, we need that methylated folate in order to help convert homocysteine to methionine. And so it's an example where the cofactor there is low. If you have an upregulated glycolysis and a low TCA cycle, this is a crucial junction point. And so if this is altered, which you'll frequently see, most famously, because we need oxygen for the whole glycolysis to go through the TCA and then oxphos, you'll see lactic acid accumulating. But this is indicative of NADH accumulation, high ATP levels. There could be enzyme deficiency or inhibition, mitochondrial dysfunction, which again could lead to metabolic syndrome. And this is also seen in aging as well. You see glycolysis is higher and the TCA cycle is going to be a little bit lower. So what are some subjective assessments in metabolomics? So these are just general questions you want to assess within a metabolomics visit. You want to be assessing energy, probably the most obvious one. What's energy upon waking? What's energy throughout the day? Does someone experience post-meal fatigue? You want to be assessing body composition. Has somebody had recent weight gain or weight loss, increase in waist circumference? Dietary assessments, all those ones, a 24-hour intake, diary, diet recall, dietary preferences, allergies, sensitivities, what supplements they're taking. Physical activity level. What is the frequency duration of exercise? Are they sedentary? What are their lifestyle habits? Sleep assessments. So how much sleep are they getting? Are they well rested? Do they sleep well? Do they wake up feeling tired? How long does it take for them to fall asleep? You can include biometrics in all of these as well. There's that add-on component to multiomics where you're also incorporating all that biometric data. So family history as well. So does somebody have a family history of diabetes, heart disease, high blood pressure, high cholesterol, or metabolic syndrome? And then obviously, do they have a current history of metabolic disease of any of those things? Objective assessments in metabolomics. So body composition, body mass index, which is BMI, waist to hip ratio, WHR, bioimpedance analysis. I'll talk about that in a little bit. Blood pressure, temperature, respiratory rate, pulse, and a nutrition-focused physical exam. Nutritional deficiencies or excess in some cases is going to disrupt metabolic pathways. And by doing a sufficient nutrition exam, you may be able to uncover those without even doing a metabolomics test, although the metabolomics test can further confirm your physical exam suspicions. And so one thing when we're looking at body composition, where is fat deposited? Having excess fat is not necessarily a bad thing, but it's the visceral fat, which is primarily more inflammatory, more associated with mitochondrial dysfunction and systemic inflammation. And so it's this excess visceral fat around the midsection, which is particularly problematic. So that's one thing to be looking out for. There's something called metabolically healthy obesity and metabolically unhealthy obesity. And it's the difference between having more subcutaneous fat than visceral fat in these metabolic healthy individuals versus these metabolically unhealthy individuals. You'll see increased visceral fat. And so it's a big differentiator between somebody who might be overweight, but they have normal insulin sensitivity, normal glycemia, normal blood pressure, versus somebody metabolically unhealthy, they have resistant insulin resistance, type 2 diabetes, and some other risk factors there. Of course, BMI, we already know what that is. This is looking at their weight over their height, and there's a chart based on where their weight and height is, what is too high or too low. There is something called a metabolic BMI. So people took BMIs of individuals, and then they looked at metabolites for these different pathways here, nucleotide metabolites, amino acids, lipids, and they were able to predict or calculate a metabolic BMI with these individuals to predict their risks here. And they found that people with a higher metabolic BMI had an increased risk of insulin resistance, genetic risks, and other cardiac events. So it's kind of interesting. Waist to hip ratio, essentially, you don't want your waist to be too much bigger than your hips. This will differ between men and women. For women, being above 0.85 is at high risk. It's more apple shaped. And then for men, anything above 1 is considered high risk for the waist to hip ratio. Bioimpedance analysis, if anyone has ever heard of this, it's essentially a device. It's these like metal plates. Some of them have where you stand on it barefoot and then you hold these metal paddles in your hands and it shoots a electrical signal through you. And this signal that is going through you is seeing what it hits. So it's looking at how much water, how much fat, how much muscle is it hitting. And from that information, it's calculating these things, how much fat-free mass, body cell mass, total body water, intracellular water, and so other things. There's a lot of these on the market. From my understanding is between devices, it's probably not that accurate, but if you stick with one device, it's mostly accurate. So just make sure you're not, trying a bunch of different devices. But it seems that these have gotten more reliable over the last several years. Blood pressure. So if somebody has high blood pressure, it's indicative of some type of metabolic dysfunction, especially endothelial dysfunction. And so that's not a good thing. That's something to measure. to extrapolate how much metabolic dysfunction is occurring. There's something called metabolic hypertension that is occurring due to this metabolic dysfunction. One thing to note, too low blood pressure is also not good. If you look at risk of cardiovascular disease and metabolic syndrome, you don't want to be too low. Everyone talks about too high blood pressure being a bad thing, and yes, it's concerning, but don't be on too low end either. There is temperature. And so there's like significant amount of people, if you're not measuring your temperature with your patients, a lot of people are not 98.6, they're like 96. They're like running low. That is their metabolic rate is low. They're just not metabolically efficient. And so you're going to see some health effects of having mild hypothermia. But it is associated with metabolic dysfunction, so it's something to keep in mind there. What is contributing to temperature? It's a lot of these processes here. So oxidative processes are going to be contributing percent to our metabolic rate. Mitochondrial proton leaking in our mitochondria, 20%. Sodium, potassium, ATP ACEs. This is where having mineral sufficiency is important. Protein synthesis contributing to this, gluconeogenesis, and then these various other processes that are ATP consuming. But this is what's generating heat within our bodies. Respiratory rate, essentially, if it's too high or too low, that could be associated with metabolic dysfunction, particularly metabolic acidosis, which the rate has altered the pH negatively, or metabolic alkalosis, where the blood pH is too high. So that's another thing to measure. Again, if you're using a biometric device, this is likely being measured, but it's something else to keep in mind. Pulse. And so there is a distribution of what a healthy pulse is in men and women. One thing to know is as we get higher in our quartiles in terms of our pulse, the higher the resting pulse, the higher the risk of developing metabolic syndrome is. And you see this trend in both men and women, especially in men. Nutritional assessment. So basically everything on the exterior you can assess. So you could look at the hair, you could look at someone's ability to smell, looking within the mouth, taste, tongue, teeth, gums. You look at one's skin, one's nails. You could do vibratory sensation to see how that's impacted. There is a lot of content here. I did a couple webinars on this. So if you are interested in going over those, looking at those webinars, those have already been recorded. We could talk about that after this, but it's especially important to do a nutritional focused physical exam. So within a metabolomics visit, some goals: you want to collect and track the subjective and objective information I just mentioned. You want to reaffirm the goals of the patient practitioner performing the metabolomics assessment. And so what has been their baseline health assessment? Is it condition specific, or is it just general health optimization? You want to establish a level of understanding of metabolomics with the patient. And so do they know what it is? Do they know what metabolites are? Have they done metabolite testing before, or metabolomics testing before? Most people think it's genetic testing. Genetic testing is the risk of metabolic dysfunction or metabolite dysfunction. You could test MTHFR. I just say that one because that's the one most people know. MTHFR is one of the key gene that makes methylated folate, and people could have risk variants in it, and then it leads to dysfunction, the methylation cycle. But how do you actually know your methylation cycle is dysfunctional? And there's symptoms that you could do, again, the subjective objective assessments, but the actual output of that would be the metabolite shifts in homocysteine, shifts in methionine, and there's actually a bunch of other metabolites part of methylation. So it's the actuality of genetic risk variants. And you could actually figure out what genetic risk variants are present by just looking at the metabolome. In an ideal world, I like both information, but I'd say that's one of the main confusions I see amongst, well, not just patients, but practitioners as well, confusing a metabolomic test with a genetic test. You want to set expectations, and so let them know what role metabolomics plays in their healthcare. What are the benefits and limitations of metabolomics, which I'll cover in a second. The health assessments and protocol identification that metabolomics is helping to elucidate. And you want to determine the depth of the review. So within metabolomics, there's all these pathways, and I just mentioned biochemistry. There's ways to simplify this, which I'll talk about in a little bit. I think it comes down to the educational standpoint of your patient. Some people know metabolites, and That's what they're looking for in a metabolomics test. Some people don't at all. They just want to know how their metabolome or their labs are impacting their health. And so it's key to make that distinction. So some benefits, limitations, and so some benefits of metabolomics provides an in-depth look at pathways occurring in the human body. It's more personalized health assessments. It can serve to improve clinical strategies in terms of disease assessment, health promotion, diet, lifestyle, and supplement intervention recommendations. It can assess the impact of environment on our metabolic pathways. And unlike our genome, which your genetic expression will change, but the genetic sequence is not going to change for the most part. And so you can retest to see how there are shifts in the metabolome that are occurring. Some limitations of metabolomics. And so usually when you do a metabolomic test, it's one sample at a time. Usually it's either blood or stool or urine. So there's that limitation. There might be changes in one or the other. The timing of the sample collection matters. As I showed earlier, the metabolome is going to express in different times in different parts of the body. There are confounding variables that will cause erroneous health associations, and so you want to make sure you account for those when looking at metabolomics. The depths of analysis is going to be dependent on evaluating accurate health information, and so when you do metabolomic testing, baseline health information is collected, and so you have to make sure you're collecting the proper information. For example, if somebody's on certain supplements, that's going to affect their metabolome, so you want to make sure you're accounting for things of that nature. And then you have to look at metabolomic information within the entire clinical picture. You don't want to just do metabolomics and completely ignore all the subjective and objective things. You want to make sure that it's all making sense, because then it's kind of irresponsible if you do it just the other way. So there is the artificial intelligence machine learning aspect of this. And when you're looking at collections of metabolites and making associations between metabolites, the data intensivity gets a little bit difficult for a human to grasp. So you might look at one metabolite and associate with a health outcome. Say you look at LDL, and we know if it's elevated. Well, I know that's probably up for debate. Some people are fine with LDL, but let's just say, generally speaking, if LDL is elevated, we know that that's possibly increasing the risk of cholesterol. When you look at the entire metabolome, there's trends. And so let's say these 5 metabolites are high, these 5 metabolites are low. When you look at the trend, what are the health associations with that? This gets to a level of complexity where you need to have some level of artificial intelligence to make those connections. And then machine learning, which is kind of from the data and from the experiences that it's presented, can learn from these data sets. Okay, when these metabolites are like this, and when these metabolites are like this person is presenting with this. And so that's how machine learning is working. So that's how metabolomics is best working. You get all the information possible from the individual sources of metabolites. What disorders are they possibly do they have? And you do the metabolomics testing. Usually it's mass spec. So, and there's GC-MS, GC mass spec, gas chromatography, or liquid chromatography. It just depends on the how they want to process the samples. And then the machine learning and data analytics occur where they're looking at those associations and making predictions based on the metabolites. So this is an interesting thing. So digital twinning, essentially, you have the entire history of the individual. You have their symptoms, you have their health history, their background, their previous diagnoses, supplements they're taking, everything. How do we know how we could favorably impact them? So what can be done is a digital twin of that individual can be created. So we make a copy of that individual and we see how can we shift the metabolome in that individual if we were to do one intervention. So let's say we do CoQ10. So we expose that digital twin to that to CoQ10, and we can calculate how that's going to shift their metabolome. And so that's one intervention. Okay, let's now add carnitine. Let's see how that's going to further alter this digital twin in terms of what we're predicting. And so this can be done exponentially many times over in order to look at what can we possibly do to create the most beneficial outcomes for this individual based on their metabolome and based on this upfront information. And so what this graphically looks like is this. And so this is the process of that happening. We are each one of these, this is a health score that is created from the individual, from the metabolome. I'll cover that in a second. Actually, do I cover that next? I don't. Because there's a couple of interesting studies doing health scores from metabolites for metabolic syndrome. So you could create a health score essentially for different areas of health, let's say insulin sensitivity or cardiovascular disease risk. And so the way the metabolites look attribute a health score. That health score is the metric you can use to see how those interventions are shifting. So for example, here's their health score. Looks like it's a 50. We do one intervention and it bumps up a little bit. And then we do another intervention and it bumps up. These are other, let's say, failed digital twins where the interventions that were suggested just were not actually improving the health score, that was decreasing. And so we could find the right recipe by looking at what protocol, diet, lifestyle, nutrition, medication, supplements, what have you, will have the most favorable impact on the health score over time. And so this is how artificial intelligence can help us. Otherwise, it's kind of guessing. It's maybe a little bit biased. Maybe there's some, I certainly as a practitioner, before I was using technology like this, or some supplements I liked, some supplements I didn't like. So that's the thing is when you use artificial intelligence, it makes it a little more objective in your assessments. And when you give it enough data, you could be more precise. I mean, this is what I'm mentioning here is precision medicine. So it's using those inputs. I know I'm just talking about this from the aspect of metabolomics. This realized is looking at the entire metabolome. So if you had someone's genome, someone's metabolome, someone's microbiome, looking at the biometric data and their health history, that would be the ultimate data compendium to figure out precisely what interventions we could do that would most favorably have an impact and what interventions we might want to avoid. So that's digital twinning as an add-on to select the interventions from the information gathered from the metabolome. And I know that's a bit of a complex thing, so we'll have an opportunity to discuss that after because I'm sure I'll have questions about it. But digital twinning is actually a newer thing that's being done in the health industry. I believe, well, I don't know if I can name companies. There's a certain airplane company that originally developed it, and now people are adopting it to health because they're like, oh, that could actually work pretty well for health purposes. All right, metabolic syndrome. So this is essentially a cluster of health conditions that can occur together: high blood pressure, high blood sugar, excess belly fat, abnormal cholesterol, and this is associated with risk of numerous different diseases: cardiovascular disease, type 2 diabetes, stroke, Alzheimer's, and various others. It's characterized by insulin resistance, oxidative stress, and inflammation. If we look, okay, well, this is the prevalence of metabolic syndrome. Some areas are pretty good: Washington, California, Colorado. And then we have some not so good areas that have a very high rate of metabolic syndrome. I mean, in some cases, more than half the population have metabolic syndrome. So it's actually not good. This data is from 2023, and it does not seem to be going away. Traditional diagnosis, we're looking at waist circumference, triglyceride levels, HDL, blood pressure, fasting glucose, and 3 of any of these 5 features is diagnostic of metabolic syndrome. We could also look at other things that can be co-occurring with this, such as microalbuminemia in the kidney, elevated uric acid, other types of blood glucose parameters, such as 2-hour fasting blood glucose or oral glucose tolerance test. So there's other tests that can indicate that there's metabolic dysfunction occurring, and it could further itself to the diagnosis of metabolic syndrome. When you look at the pathogenesis of metabolic syndrome, as I mentioned, there's inflammation, so tumor necrosis factor, interleukin-6, CRP. These are elevated. We have excess free fatty acids, high leptin, low adiponectin, increased ROS, increased renin, angiotensin, aldosterone system activation. This is resulting in these not so good things: chronic inflammation, and insulin resistance. The microbiome will impact our metabolic syndrome risk. Elevated levels of Firmicutes and Bacteroides, low bifido and lactobacilli, and some other ones I'll talk about in a little bit, such as Akkermansia. This is associated with dysbiosis, some metabolites that are high from these more westernized diets: choline, carnitine, trimethylamine oxide. I think our microbiome will impact how these are being metabolized. If you have a healthy microbiome, I don't think these are as much of a concern, but most people that are eating a more westernized diet don't have a good microbiome, so these become more problemsome. You'll see elevated pro-inflammatory molecules such as LPSs, certain tryptophan metabolites, leading to endotoxemia, inflammation, and subsequently metabolic syndrome. Whereas in a more healthy gut microbiome state where you have elevated levels of bifido and lactobacilli on something such as a Mediterranean diet, you'll see increased short-chain fatty acids, which will decrease inflammation and subsequently decrease our metabolic syndrome risk. I mentioned the mitochondria was crucial for metabolic dysfunction. Lots of different things can mess up our mitochondria. High caloric intake, mutations in our mitochondrial DNA, excuse me, environmental toxins, homeostatic imbalances, glucolipotoxicity, which is going to alter our mitochondrial endoplasmic reticulum and interactions, all leading to mitochondrial dysfunction. And what that specifically means is our electron transport chain is not going to be transporting electrons properly to do the respiration process, which makes ATP. It's going to be leaky, so you're going to have ROS leaking out, which is going to lead to lots of not good things. It's going to alter insulin signaling, lead to cell death, increase oxidation, and ultimately will lead to insulin resistance, elevated blood glucose levels, which is going to increase our picture of metabolic syndrome. So you want to have metabolic balance. And so in a more balanced state, our nutrition is good, our environment is good, our genetics, if we have risk variants, are not hurting us too much. We want to be avoiding food allergens, have low GI tract inflammation. We want to have balance in our metabolism, and oftentimes with something like metabolic syndrome, there's great imbalances. So what metabolites are associated with metabolic syndrome? So there are many studies on this topic. Just to highlight a couple, and if you've not looked at metabolic studies, they look like this. There's lots of dots, there's lots of points, there's molecules you might have never heard of, and then there's molecules you have heard of. Fucose, which is from brown seaweed, methylmalonic acid, you'll see changes in these. Methylmalonic acid, if anyone's familiar with, is associated with B12 metabolism. You'll see low creatine, pyroglutamic acid will be altered. This is associated with glutathione metabolism. And so you'll see certain changes that are occurring in metabolic syndrome. When you look at pathways, arginine and proline metabolism, oftentimes with most diseases, arginine metabolism is thrown off for whatever reason. I believe this is supposed to say glutathione metabolism. I don't know why there's an M there, but glutathione metabolism is going to be altered. Taurine is part of the glutathione metabolism pathway, and so it makes sense that we would also see that this is actually way altered. And so you'll see those things dysfunctional. What is also done in these studies, not just associating with metabolic syndrome, but the hallmarks of metabolic syndrome. And so we could also look at these individual things. So waist circumference, what metabolites are associated with that, BMI, metabolites that are associated with that, triglycerides. So it's interesting, this fucose. This is brown seaweed is supposed to be a metabolic activator, and this is very low. Not only metabolic activator, it helps activate our body's ability to burn fatty acids. You look at these people with elevated triglycerides, this fucose is very low. So it makes sense. You could look at classes of pathways, and so obviously within metabolic syndrome, you're going to see free fatty acid metabolites, alterations there. Carnitines are crucial for our body to burn fats as a fuel, so it makes sense you'll see dysregulation there in that pathway. Glycerophospholipids, there's all different types of these, and so you'll see these dysregulated in various different diseases. It seems like when the body is ill, it just doesn't process these glycerophospholipids properly. Some of them we've heard of, like phosphatidylcholine, phosphatidylinositol. Whoops. Amino acids, serine, glycine, you often see those dysregulated, arginine as well, like I mentioned, and TCA cycle, of course, ketone body oxidation, you'll see alterations in these metabolites as well. So as I mentioned, you can create a health score. So looking at the metabolites, you can create a score, and based on those scores, you could put people into quartiles. And so you can score dyslipidemia, score blood pressure, score fasting should be blood glucose, and just metabolic syndrome in general. And what's nice about this is it's difficult to look at just a bunch of metabolites and those pathways, but if you could attribute a score, it makes it clinically useful because as a clinician, you're busy and it's simpler to look at a score. If somebody comes in to your clinic and you do a metabolomics test and they have like a 40% metabolism or metabolic syndrome score, you do work with them, you see the score go up, it's looking good. Now, and from that, you could look into what is going into the score, and then that's where you could get into the nitty-gritty details of looking at individual metabolites, how they are shifting, contributing to changes in the score. But the score is helpful nonetheless. And so with that information, as I mentioned, we can gather a lot of information on somebody to do the data analysis. Not just our metabolome, we could look at the microbiome, we could look at the proteome, we could look at non-omic factors such as socioeconomic status, age, sleep, and stress. And we could use that information with AI to make more personalized interventions and recommendations. So traditionally, what is recommended for metabolic syndrome? Lifestyle promotion, which is going to vary based on the person that's administering it. Medications, of course, statins, fibrates, metformin, acarbose, glucan-like peptide. It's an infamous one now. So lots of different interventions are traditionally recommended for it. We could exercise. It makes a huge impact on metabolic syndrome risk. When we look at exercise, we could look at changes in the metabolome and how that may be impacting our health. As I mentioned before, carnitines are very important for fatty acid oxidation. And so you'll see changes with those when an individual is exercising. This one, indole lactic acid, is a very anti-inflammatory molecule. You'll see shifts there. So you'll see various shifts from exercising on the metabolome that's going to help with metabolic dysfunction risk. There's many different ones that have been studied. This is actually one of my favorite ones, BAIBA. This is something you could actually supplement with if you want, but if you exercise, your muscles will make it. And it has many different actions. It increases adipose tissue browning, which is a good one. It has brain benefits, increases fatty acid oxidation. Also, alpha-ketoglutarate is another one that's been associated with anti-aging benefits, has numerous benefits from exercise on muscle and brain and bone loss. There's many different diets. Of course, Mediterranean diet is the most well-studied one for metabolic syndrome, but there's other ones that have been shown to be beneficial, DASH diet, plant-based diets, and so that could varies based on the starting point of the individual. Probiotics, many different ones, particularly in humans, what have been studied is Lactobacillus gasseri. These specific strains have been associated with decreased metabolic syndrome risk. That's in humans. In rats, there's been several others that have been looked at. Those were probiotics. These are prebiotics. Be careful with prebiotics in individuals with gut issues, especially SIBO. Though if their gut can handle them, they've been associated with various different benefits for decreasing metabolic syndrome risk, mainly through helping increase these probiotic bacteria that we know are beneficial in metabolic syndrome. Postbiotics, I really like these, especially bacteriocins. It helps answer the question, if somebody's been on antibiotics their whole life, is it contributing to them getting sick more often? They have cleared their body's ability to make these antibacterial, antiviral substances because we've killed them off. But if we had a healthy microbiome, they would make it for us. Plasmalogens is another really interesting one. It has particularly cognitive benefits. Our microbiome will make it. And many other different things, especially short-chain fatty acids is the most well-studied one, different vitamins, different enzymes, so lots of different things made from a healthy microbiome that will help with our risk. Akkermansia, it's probably one of the most popular probiotics over the last several years. Numerous different benefits on metabolic syndrome. You'll find if you test this on individuals that are metabolically unhealthy, you'll see low levels of it or undetectable. Sometimes it's not in them at all. So different foods can help increase the growth of this, such as cranberry and pomegranate. And I've seen some companies actually marketing this as a supplement. It has numerous different benefits on the metabolism. And of course, bioactives. So from the diet, all these different things here, coffee, green tea, curcumin, they have been well studied to have numerous different impacts on various facets of metabolism, and especially those markers associated with metabolic syndrome. We can shift those in order to help decrease our risk and move us in the right direction. Bergamot is one of my favorite ones for hypercholesterolemia, hypertriglyceridemia. Cinnamon's another really good one. So just through diet and spices and herbs, there's a lot we could do to move the needle. All right, so some takeaways. Metabolomics is a study of molecules, metabolites, and their health and disease associations. Metabolomics provides key insights into normal versus pathological metabolism. In practice, we should consider a metabolic-centered subjective and objective assessment in conjunction with analyzing one's metabolome, so don't do one without the other. Metabolic syndrome is associated with dysfunctional metabolism. And so pathways that you could look at or that have been found to be dysfunctional with metabolic syndrome include arginine, proline, glutathione, and taurine metabolism. Those other fatty acid pathways that I mentioned will be dysregulated. If you look at specific metabolites, methylmalonic acid, like I mentioned, pyroglutamic acid, phosphatidylcholine, and indole amino acids will be altered in metabolic syndrome. And you could use digital twinning as an intervention to help select appropriate interventions to improve metabolic health risk scores to decrease metabolic syndrome risk. And with that, I thank you for attending, and I will take questions at this time.
Okay. That was great. Okay.